This video is called Protein Synthesis 101. So it's the first day of notes about protein synthesis. And, we've gone through DNA, DNA replication, DNA structure. We've talked then about mitosis and meiosis. So you've gotten a real good kind of a background on what DNA does. But one of the things you may not realize DNA does, besides having all the information how to make another you and how to make another organism, every single cell that has a nucleus has all the DNA in order to make another organism, just like the one that it came from. In addition to that, the DNA carries all the information needed for the day-to-day operation of the cell. Most of this all involves proteins. And so we're going to talk about protein synthesis today. And there's like big picture of this. There's two main points that you need to remember. And one is transcription, writing of the message happens in the nucleus. Translation, of course, we can't translate a message until it's written. So you need to remember transcription and then translation. It's also alphabetical, so one of those will help you remember. So trans-C and then trans-L. Translation happens out at the ribosomes, and that's where proteins are made from the information that comes from the nucleus. So, protein synthesis, two main parts, transcription and translation. I would make a flashcard that had those pieces of information for sure and kind of a definition of each. Back when we talked about DNA replicating, there was an enzyme called DNA polymerase that you may or may not remember, but the DNA polymerase was an enzyme that unzipped the DNA so we could make a new copy on each of the strands, and then we'd have two complete copies of DNA. So the replication is one of the processes that happens with DNA, but transcription and translation are another process, and this helps make proteins that will be used by the cell and also by the body. So you want to make sure these words that are in red, make sure that you know the definitions of them at least. You might want to make a flashcard of them. There's lots of index cards that you can use. So the DNA polymerase was what helped make the copy of DNA. RNA polymerase is what unzips the DNA to make a new molecule that we're going to call messenger RNA. Now that's real appropriate because it takes the message out of the nucleus to the ribosome, and RNA is similar to DNA in a lot of ways, except it's a single strand. It has ribose instead of deoxyribose, and one of the bases is different. Wherever you would see T in DNA, you're going to see U for uracil in RNA. So, if you're ever trying to make the code and you write the code out and you've got T's in RNA, get rid of them, erase them, and put U's, because uracil replaces thymine in RNA. So, you should know those differences too. Single-stranded, ribose instead of deoxyribose, and uracil instead of thymine. So, out at the ribosome, this messenger carries the message from the nucleus. You don't want to take the original plans out of the nucleus because that's the original. And if you damage those, we wouldn't have a copy for how the cell was supposed to get along with its life. So we make a copy in messenger RNA, send the message through the pores of the nucleus out to the ribosomes. The ribosomes are outside the nucleus, and they do the process out there, which is translation, and they make the proteins in the same order the code says that they needed to from the DNA. So, this is a look at transcription, and it's just a picture. Yours is small, but you might be able to see here that A makes a U, T makes an A, G makes a C, C makes a G, T makes an A makes a U. This is the messenger RNA. So this top strand, that's the DNA. The bottom strand is the mRNA, and it'll be a long strand. It's a lot longer than this little strand that you see here. But once it gets to a special 3-letter codon called a stop codon, there's no code for that, and that means that it's a blank, so the messenger RNA drops off and it goes out through the pore out to the ribosome. So these are called ribonucleotides. Remember, the little bases are one of the parts on a nucleotide. You've got phosphate, sugar, and base. So these are the ribonucleotides that are assembling in place, and RNA polymerase does that. So this is also another picture of translation out at the ribosome. With the message now has left the nucleus, and it goes out to the ribosome. And here's the messenger RNA, and the messenger RNA goes through the ribosome, and it's got a 3' and a 5' end. They are different. That's not a really important thing to know at this level, but it goes through from 3' to 5'. And as the message goes through, we have this T-shaped RNA called tRNA, or transfer RNA, that brings the correct amino acid that corresponds to the code that came from the DNA. So the order of the DNA tells what the order of the amino acids are, and the order of the amino acids determine what the protein does, because it determines the shape of the protein. So you can see that we've got glycine, serine, glucine, valine, lysine, cysteine. These are all amino acids, and if you look up, there are about 20 or so amino acids that we have that grow and form into a protein. A protein has 50 to thousands of amino acids, so it's a really, really big molecule, but it's made of amino acids. Amino acids are the building block for proteins. Again, this happens at the ribosome. So we're kind of getting big picture today on this, so I want to, as much as possible, hit the big details. So there are two main areas that we're going to talk about with genetic code. First of all, remember that A copies to U in mRNA, so A DNA would produce a U in the mRNA code. And the rest of them are all the same as what you've learned before. T goes to A, G goes to C, and C goes to G. So if you're given a strand of DNA and it tells you to put the corresponding complementary DNA, well, it's just G to C and T to A. Wherever there's a T, there's an A in the DNA code. Wherever there's an A, there's a T. And if you can remember, the round letters go together. G and C are both round. And the flat-sided letters, A and T, have flat sides. Those go together. But in RNA, now that's in DNA, in RNA, you do that, but when you get finished, in the RNA message, any T is not going to be T, it's going to be U. Always replace the U into where a T would normally go in RNA code. There's no T's, no thymine in RNA. So that is the code that every organism uses, from acorns to mosquitoes to humans. Every organism has the same genetic DNA code. So this tells you a little bit about the DNA. It's double helix, A across from T, G across from C. This tells you a little bit about RNA, where we have G and C match, T and A match, C and G, but across from A, this triplet, across from that A is a U. So ACA corresponds to UGU on the mRNA. The top part's the DNA, the bottom part is the mRNA. And only one of the strands of the DNA has the code that's copied to mRNA. The other one is just there as a complement, and it's used during replication, but not during transcription. So here's the other part of the genetic code, and one that you'll probably be asked to use maybe on the final or on the final review. Notice that it has the letters here, UUU, UUC, both of those correspond or code for phenylalanine. That is an amino acid. UUA or UUG code for leucine. So you can see that there are about 20 different amino acids here, and one of them, AUG, the methionine, that's the start code. That's like a capital letter in a sentence. It says, here's the beginning of the gene. This is the beginning of the message. And then we've got a couple that correspond to the stop code. There's 3 of them actually. UAA, UAG, and UGA say stop, and they don't code for an amino acid. So at the end of a long message, there'll be a blank where there's no amino acid, and that causes the protein to fall free, and it goes out to where it needs to go. So these are, just like words mean things in languages, these triplets mean specific amino acids. And this is the table that has that coding. You do not have to memorize this table. If you're asked questions about it, you'll be given one of these as a possibly useful page. But you should look over it and be able to use the code that's here. You should be able to see, let's see, what does CCU code for? Well, CCU is right here. Its first letter is C, second letter is C, and the third letter is U. Okay, so CCU. That's how you, first letter, second letter, third letter, and you find it. Or you could just look through the chart till you find it. It takes a little longer. And that codes for proline. So that would be the genetic code, kind of the big end genetic code that determines which amino acids are going to appear in proteins based upon the message that comes from the DNA. So one more time through for, I think, the important big picture. Make sure you know that transcription with the C occurs first in the nucleus at the DNA. DNA splits, writes a message in messenger RNA. Messenger RNA goes out of the nucleus through pores, out to the ribosome, and the ribosome is where protein synthesis takes place. That's where we make proteins, and they're made in the order that the DNA coded for and that passed on in the messenger RNA. So that message goes out to the ribosome. Transfer RNA brings in the correct amino acid. It has a 3-letter triplet that's an anticodon that matches the codon, and it brings the right amino acid for that 3-letter code. And then it puts the amino acid in, it's chained to the last one, and then it moves over one, and the next one comes in, and the next, and the next, and the next. And it's like a chain of sausages. By the time you're finished, they keep adding on until you get to a stop code. So that's the big picture. Make sure that you know the big picture, and the more of the details you can learn, the better. If you need to ask questions, come down and ask questions. Don't be a stranger to tutorials. Work on your poster. You're going to have a poster that kind of gives the big picture of protein synthesis. Good luck.